Welcome back, my dear students. So in the last video we had started with immunity. So the types of immunity, then antigens and antibodies, we had studied about all that, and antigen and antibody reaction. So I told in this video we will be starting with the cell mediated immunity. Okay, that means cells that are present in immune system and how they provide immunity to the body. Okay. So the cells of immune system, there are basically two types of cells present in the body, or which act as cells of the immune system. One is antigen presenting cells. Okay, antigen presenting cells or APCs. These are non-phagocytic. They don't act on the antigen or the foreign body. They only help in presenting these antigens to the immune cells, the other immune cells, which will act on it or destroy it. Okay, so these antigen presenting cells, and the other is lymphocytes. So there are two types, B lymphocytes and T lymphocytes. So this will be a little confusing. You have to listen carefully, then only you will understand. Okay, so how does this antigen presenting cells help, or how does it work? So they ingest and break down the foreign antigens. They break the foreign antigens and display their internal peptide fragments that are present, that will be displayed on the cell surface. So automatically what will happen, that will help in the recognition. Yes, so this actually helps in the recognition of these antigens by the other lymphocytes. Okay, so these MHCs, which are called major histocompatibility complex, so there are different groups of that. So out of which MHC2 is present on the surface of cell surface of APCs. Okay, on these MHCs combine to the antigen. Yes, so antigen presenting this MHCs will combine and form a complex. So now this complex is identified by the T cells. Clear? So these T cells, when they identify, they get activated. Which T cells? There is a group of T cells called helper T cells. Okay, this helper T cells produces clones of itself, of the same helper T cells, and secretes cytokines. Okay, so these cytokines will activate the B cells and cytotoxic T cells. That is another group of T cells. So cytotoxic T cells will help in breaking or cell lysis of the antigens, and B cells on the other hand will help in the production of antibodies. We have already seen that B lymphocytes or B cells help in the production of antibodies. Yes or no? So this is the process that is happening. They ingest and break down foreign antigens and display the peptides on the surface. MHC, which forms a complex, this helps in the identification. So just know what happens, what is the function of APC. It helps, or it combines with the antigen to form a complex. So this complex is identified by the T cells. Okay, so that is antigen presenting cells. Then after that, the process, whatever is happening, immune system, the other cells will take care. Either it will be engulfed or it will be lysed. The cell will be lysed. Next is lymphocytes. Okay, so lymphocytes, we have all heard about lymphocytes. These are types of WBCs. Yes, these are the main cells of the immune system. So they are form the cellular elements of the immune system. Okay, there are two types, B lymphocytes and T lymphocytes. So in general, the origin of these B and T lymphocytes, how does this happen or where does this take place, we'll see. So although these B and T lymphocytes are present in the lymphoid organs or lymphoid tissues, they develop or originate in the bone marrow along with the other blood cells. Okay. So this formation of these blood cells is called hematopoiesis. So in this itself, the lymphocytes are also produced. In hematopoiesis, how the production takes place, that is through pluripotent stem cells. So these stem cells have the ability to develop into different types of blood cells. Okay, so pluripotent stem cells, that is also gives rise to two kinds of stem cells. One is myeloid stem cells and the other is lymphoid stem cells. Myeloid will give rise to the other types of blood vessels, cells, and lymphoid stem cells gives rise to lymphocytes. Okay, B and T lymphocytes. Alright, so their development and maturation, based on that and their activity, they are classified B and T lymphocytes, also known as B cells or T cells. So B cells complete their development in the bone marrow and then they are carried to the lymphatic organs or lymphoid organs. Okay. Whereas T lymphocytes, they develop from the stem cells. They develop from the stem cells and then they migrate to the thymus. Okay, these stem cells from which T cells develop, they migrate to thymus. Okay, there it develops and matures. So antigen receptors on the surface for recognizing specific antigens. So in thymus, they contain or they require antigen receptors. So antigen receptors means which can identify for the recognition of the specific antigens. Okay, that is the development. How does it act or how does it function, the mode of action? So mode of action of B lymphocytes is, B lymphocytes are sensitized either directly by the antigens or by T cells. So I had told that the T cells, when they get activated by the complex, that will activate B cells or will sensitize B cells. Yes, so they are either sensitized by antigens or T cells. Then after they are sensitized, they enlarge and divide to form plasma cells. Okay, so these activated B lymphocytes, they will enlarge and undergo division to form plasma cells. These plasma cells contain or produce antibodies, protein substances called antibodies against these particular antigens. Okay, and these antibodies provide humoral immunity to the body. Okay, so they circulate in the blood and the lymph to fight against these specific antigens. Clear? So that is the mechanism or the mode of action of B lymphocytes. So how does now T lymphocytes, we'll see. It identifies a specific antigen. Okay, either by the help of APC or otherwise also it can, if antigens enter, they will be able to identify the antigen. Okay, so here also they are antigen specific, so they will recognize a specific antigen and this will stimulate the T cell to produce different types of T cells. Okay, so based on the type of antigen that is produced, the different types of T cells will be produced. Clear? So the type of antigen which it is encountering, based on that it produces a clone. So I had told that the T cells will produce a clone. It can produce clone of itself. There are 4 types of T cells based on that. One is helper T cells. So helper T cells, what do? They again help in the activation of B cells and other cytotoxic cells. Then we have killer cells, killer T cells, also known as cytotoxic T cells. Then suppressor T cells and memory T cells. These are the 4 types of T cells. Okay, so we will see, we'll see the how it functions. So here you can see the activation. Okay, so this is the antigen and the MHC antigen complex that has been identified by the T lymphocyte. Yes, T lymphocyte has identified and attached. So based on what kind of antigen it is, it will produce clones. Cytotoxic cell T cell, memory T cell, suppressor T cell, or helper T cell. Okay, what are the functions of each? Cytotoxic, it is to break down or lyse the cell completely. So how that happens, I will explain. Before that, memory T cells, these are sensitized by antigens and retain sensitization for the future. So first, when it is attacked by the antigen for the first time, it gets sensitized. So if suppose this T lymphocyte identifies this antigen as an already or a second as its second encounter or a third encounter, then it will produce memory T cells. So memory T cells will know that what has to be done because it is sensitized, has already been encountered with this particular antigen, and then it will act accordingly. Okay, now suppressor T cells, they inhibit the immune response by releasing cytokines that suppress activity of other T cells and B cells. Okay, they suppress the activity. Next is helper T cells. So I told they help in presenting or activating the other cells, B cells and cytotoxic T cells. Okay, now see here, this is cytotoxic T cells or killer T cells. What happens? This is the cytotoxic T cells. Okay, it has attached to the antigen, foreign antigen. The MHC is there, MHC major histocompatibility complex. Now these cytotoxic T cells, they produce perforins. What are perforins? They are chemicals or they are substances which produce pores in the cell membrane. Okay, so when cell membrane pores are produced, so water and ions will enter, keep entering, and when water and water keeps entering, it will bulge and it will cause in the bursting of that particular cell. Okay, that means it has ruptured. So now this ruptured cell will be engulfed by the phagocytic cells. Alright, so that is how cytotoxic T cells function. Okay. Now these are some of the differences of B lymphocytes and T lymphocytes. You can go through these differences. There are many based on their function, based on their development, what happens, where they are formed, where they mature. So all those lifespan, many differences are there. So you can go through. Okay. So B lymphocytes, we know it mainly deals with humoral immunity. Here T lymphocytes, cellular immunity. Okay. Now activation of adaptive immunity. Adaptive meaning acquired immunity. So how it is, the process is happening. So the antigen is processed by antigen presenting cells. Okay, the antigen is identified and processed based on the antigen. What kind of processing has to be done? If the cell wall has to be degenerated and internal structures have to be exposed in that way, or they form a complex so that the other immune cells can identify. Okay, so the processed antigen is located on the surface of the APC. So they are located on the surface of the APC and form a complex. Now the T helper cells get activated. Yes, so now the activated T cells will activate other T cells, that is cytotoxic T cells or B cells. So B cells, they will produce antibodies, okay, or T cells, they will rupture the cell. So how the action has to be, so we know T cells are specific to each antigen. So what kind of treatment or what kind of action has to be carried on to a particular antigen, based on that the activation will occur, okay. Then they produce clones of the cells. So I told they produce clones of the cell. B cells will produce clones of the plasma cells. Yes, they'll produce plasma cells. Here T cells also, they'll produce their own clones based on their action, based on the antigen. These cells of the clone will recognize the antigen and eliminate it. Clear? So this is the process that is happening, or activation of the acquired immunity that is occurring. All right. Okay. Next is the immune responses. So based on the antigen I told, there are different actions taking place. So what kind of immune responses shown in the body? There are two types: primary immune response and secondary immune response. So primary immune response is produced by the initial contact with the antigen. So first time the body is getting being encountered with that particular antigen. That time the response will be fast, and it will decline fast. Okay, the process will be faster, and it produces both receptors and memory cells. So since first time, so it will produce memory cells, so that if there is any other encounter, these memory cells will take care. Okay, so the receptors are also formed. So in the when it is for the first time, it will produce receptors, specific receptors for specific antigens. Next is secondary immune response. In secondary immune response, it is the immune response to a subsequent encounter with the same antigen. So if there is a second encounter or more than that with the same antigen, then it is called secondary immune response. Secondary immune response is developed. It is also called booster response, and here the response is more intensified, more rapid and intensified. Okay, and this lasts longer. This immune response lasts longer than the primary immune response. So in primary immune response, we had seen that it declines fast. Yes. Okay. Now coming to the lymphoid organs. Lymphoid organs, there are two types: primary lymphoid organs and secondary lymphoid organs. Primary will be bone marrow and thymus. So bone marrow and thymus, they are the lymphoid organs where it is produced. So it is the bone marrow is the main lymphoid organ where all blood cells are produced. So here the maturation of B lymphocytes take place. Whereas in thymus, it is an organ, lymphoid organ present near the heart, beneath the breastbone. This is very large at the time of birth, but after a certain age, after puberty, it becomes small. Okay, thymus is the site for development and maturation of T lymphocytes. I had told already that it migrates along with the stem cell. It migrates with the stem cell to the thymus, where it matures and develops. So in other words, both these primary lymphoid organs, that is bone and thymus, both of them provide the environments for the proper development and maturation of the lymphocytes. Clear? Okay. Next is secondary lymphoid organs. Secondary lymphoid organs, there are spleen, lymph nodes, tonsils and malt or malt. It is also called mucosa associated lymphoid tissue. So we'll see one by one. Spleen, you might have studied a little bit about spleen in your junior classes. It is a bean shaped organ which contains the lymphocytes and phagocytes. Okay, it is a large reservoir of erythrocytes because it destroys the worn out RBCs, and it also produces RBCs in the embryonal stage. Okay, it acts as a filter of the blood as it traps the microorganism. Now coming to lymph nodes, they are small solid structures located at intervals along the lymphatic system. So along the lymphatic system, lymph nodes are present, from which the lymph vessels are develop. Okay. Now some of these lymph nodes also act as lymph glands, which produce certain substances against the microorganisms, like tonsils. Tonsil is also a lymph gland. It is also considered as a separate lymphoid organ. So these lymphoid tonsils, what they do, they protect the body from microorganisms or any pathogens by localizing the infection in itself. So it takes in the infection or traps the pathogen and prevents it from spreading to other parts of the body. It also helps in the formation of WBCs. Now coming to MALT, that is mucosa associated lymphoid tissue, it constitutes of 50% of the lymphoid tissue, and it is located in respiratory tracts, digestive tract and urinogenital tract. That means the mucosa, we know mucus secreting, where whichever tract the mucus is secreted. So mucus, I had already told mucus how they help in the protection. Yes, they trap the microorganisms. So here this lymphoid tissue is present. All right. Okay. So now we'll see the different two types of acquired immunity. We had seen the components now till now. Yes, humoral and cell mediated. This is the types of immune system or acquired immunity. One is active immunity and passive immunity. In that also there are two: naturally acquired, induced active or acquired immunity, and natural passive immunity and artificial passive immunity. Okay. So what is active immunity? Active immunity is when the it is produced by antibodies and cells that develop in the response to antigens. Okay, so that means antibodies or the reaction, immune response occurs by the cells which encounter antigens, whereas in passive immunity the immunity is provided by another source. All right. So in natural active immunity, it will develop after exposure to the antigen, and the immune response will develop. Or induced, it is when you add a little bit of the antigen to prevent the disease, like in vaccination. Vaccination, what do? We add a very small dose of the organism so that the body itself produces immunity against the disease or the microorganism. Now coming to passive immunity, what happens? One is natural. Natural passive immunity is when the immunity is provided from mother to by maternal antibodies from the mother to the child. That is an example of natural passive immunity. So through placenta the antibodies pass. So I had told that which is the antibody that can pass the placenta, if you remember, that is IgG. Yes, so IgG will provide immunity. And even in colostrum also, it contains secretory immunoglobulins, that is IgA, that will also be help in the providing passive immunity. I mean, these are the antibodies involved. Okay, so from mother to child through placenta or by breast milk, natural passive immunity is given. Otherwise, the antibodies are injected, administering the antibodies to combat infection. Okay, that is artificial passive immunity. Okay, so I hope you have understood active and passive immunity. Active is when the body itself develops. Passive when body is provided with the necessary conditions or necessary antibodies. Alright, now we'll see vaccination. So vaccination is the process of introducing a vaccine in the body to develop immune response against a particular microorganism or particular disease. Okay, how does this take place? What is the process involved? So once the body is immunized by infecting, immunization is provided to the body by infecting it with the or introducing the microorganism or the antigenic pathogen in a weakened condition, either weakened or killed into the person. Okay, now immunity develops due to the different cells of the immune system. So when the infected person when infected by the person, memory cells will develop. Okay, and or the T cells and the B cells will recognize these antigens. All right. And once these antigens are identified, the other cells of B lymphocytes will produce antibodies against these antigens, and it will induce the formation of T cells, B cells and antibodies. So that is how vaccination helps in the immunization process. Okay. All right. So types of vaccines, there are different types of vaccines. They're classified in two ways. One is based on the mode of preparation and based on method of preparation. Mode of preparation will be either it is live, that is live organism, or killed germ, toxoid, cellular fractions, or in combinations. So in live vaccines or attenuated vaccines are prepared from live, weakened or non-virulent organisms. Weakened or non-virulent. So for measles, mumps, typhoid, OPV. OPV is oral polio vaccine. So any one example you can write. Even BCG is also a live vaccine. Vaccines from killed germs. Vaccines from killed germs, we have that is the pathogen is killed and then it is entered. So that means it is not alive, but there are some things inside this microorganism which will provide or which will trigger the body to produce an immune response. So for even plague, rabies, cholera, all these vaccines are made from killed germs. Toxoids, there are certain organisms such as bacilli or for diphtheria, tetanus. The toxoids are secreted by the bacteria itself, which are harmless, but it helps in the treatment. So this is introduced into the body. Okay, for diphtheria, tetanus, this is helpful. So this provides a passive immunity. Yes, so we are providing it. The toxoids that are produced by the cell itself, we are extracting and giving it to the body. Cellular fractions, extracting cellular fractions from the microbes. Okay, for pneumonia, hepatitis, so all those are from cellular fractions. In combination is when two or three microorganisms, they combine the vaccines. So in that case, it is effective for all the specified diseases, okay, like MMR, DPT. MMR is for measles, mumps and rubella. DPT for diphtheria, pertussis and tetanus. Now coming to based on method of preparation, first generation vaccines are there, second generation and third generation. First generation is when it is produced by conventional techniques using the whole microorganism. Okay, so the whole microorganism is extracted either in the live attenuated form, killed, or inactive, it is inserted. Next is second generation, where a portion which is useful in triggering the immune system, that is extracted and it is developed by using DNA, rDNA technology. Okay, so hepatitis is an example of second generation vaccines, and third generation will be when these are chemically synthesized against the certain diseases. Okay, so that is usually for snake poison, snake venoms. They are artificially antibodies or antitoxins are prepared, antivenoms. Yes, so that is about the types of vaccines. Now coming to the disorders of immune system. So disorders of immune system, we have allergies, autoimmunity and immunodeficiency. So allergies is also known as hypersensitivity. So during any kind of allergy, the IgE type of antibodies are produced. So allergy is an exaggerated immune reaction or response by the person due to hypersensitivity of a foreign antigen. So allergen is the substance which causes this allergic reactions. Okay, so there will be many symptoms are there, like sneezing. Different people can be allergic to different things. Okay, if a person is allergic to pollen, some other people may not be allergic. Yes, some food they are allergic to, some plants they are allergic to. Yes, so that is why some even even when they come near those plants, that causes allergy. So allergy can be, symptoms can be sneezing, itching, rash, skin rashes, inflammation, swelling, watering of eyes, inability to breathe. Okay, and expression of allergic reactions. What are they? How do they express? How does it occur? So first exposure, the allergic reaction is not caused. Okay, in that first exposure, what happens? The immune system, it suppresses and it produces memory cells. Okay, so antibodies are produced and it is harmless during the first reaction. Second, or after that any kind of encounter, then it produces allergies. That time they produce IgEs and it sensitizes the body. Okay, so if you see here, what happens is, see, this is allergen, then B cells will identify, or it produces the antibodies. IgE antibodies are produced, and antibodies, so mast cell, why mast cell? Because mast cell is a APC. Okay, I had forgot to mention, APCs can be macrophages, mast cells, dendritic cells, all those are different types of APCs. Okay, so this mast cells which has the antigens attached, so this complex, antibodies also come. So mast cell and antibody complex, allergens get attached to it, and this will now bind with the allergen, and histamine is released. When histamine is released, allergic reactions due to the allergens, the histamine releases and produces the symptoms. Okay, so hay fever, common allergies are hay fever, allergic asthma. Okay, anaphylaxis, urticaria, all those are different allergies. Treatment, coming to the treatment, antihistamines are given, because histamine is that produces a immune response, that kind of immune response, the allergic immune response. So if antihistamine is given, it will subside or suppress the histamine that is produced. Okay, or steroids are given in case of severe conditions like asthma. Adrenaline is given, which reduces the symptoms of allergy. So all these medications are provided. This is the mechanism. What happens? So first exposure, nothing is happening. Yes. So they produce antibodies and memory cells. Yes. Here, second exposure, this will be ready by then when the allergen comes, and then it ruptures the mast cell to release the histamine. Okay. Autoimmunity, I had told that if at all the body fails to recognize its own cells, the self cells, differentiate between self and non-self, then this disorder arises, autoimmunity. So immune system of the body starts attacking its own cells. That is autoimmunity or autoimmune diseases. So depending on the type of cells recognized as self antigens, they are recognized as self antigens. So self antigens means they are antigens present in the body. So what the immune system of the body thinks that it is a antigen, so it has to react against it. Okay, so chronic anemia, myasthenia gravis, chronic hepatitis, Hashimoto's disease, rheumatoid arthritis, Addison's disease, all these are different types of autoimmune diseases. Alright, so you don't have to know in detail about that. Just remember that if it fails to identify the self and non-self cells. Okay, so they are recognized as self antigens. They recognize these body cells as self antigen. So suppose if RBCs act as, or they recognize these RPGs as self antigens, they will start destroying the RBCs. So what will happen? It will lead to the anemia of the person. So person will become anemic and and it will cause anemia in the person. So it can be very severe chronic anemia. So imagine all the RBCs being destroyed in the blood. Yes, so that is an autoimmune disease. So if such a condition arises where body keeps reacting against its own cells, it will lead to autoimmune disorders. Next is immunodeficiency diseases. So immunodeficiency diseases when the immune system, there is no immunity in the body. Okay, don't get confused with autoimmune and immunodeficiency. Autoimmunity means body's immune system is perfect, but it is not able to recognize which cell is what. So it is attacking on its own body cells. Whereas immunodeficiency, the immune system becomes, or immune system is reduced. So it is not able to fight against various diseases. Okay, one of the main immunodeficiency diseases, AIDS. Okay, so I am not going in detail. This is already done in the diseases part. Okay, so it is a disorder of cell mediated immune system. So reduction of number of CD4 or helper T cells. So once you know the function of helper T cells, how important it is. So if helper T cells are not there sufficient enough, then the immunity of the person will be reduced. So it inhibits the production of antibody by B cells. Yes, helper T cells was one that activates B cells and further production of antibody. So if helper T cells are not there, so antibody production by the B cells will also be reduced. Okay, so that is what happens in AIDS. So structure of virus also I have shown. It is a retrovirus and it has a single stranded RNA which has reverse transcriptase. So what happens when it enters the body? here. So when it enters the body, viral particle enters the body and it replicates inside the body, gets converted into DNA, and it combines with the DNA of the host and replicates along with the DNA. Okay, so this replication of retrovirus also you can go through how it happens, how this process takes place. Mode of transmission: sexual intercourse. If a person has AIDS or HIV positive, it can be transmitted through sexual intercourse. Transfusion of blood, shared needles, so basically blood related. And since it is present in the fluid, semen or in vaginal fluids, it also contain the virus. So that is why through sexual intercourse, transfusion of blood, shared needles, and from mother to child. That means it can be transmitted from the mother to child through breastfeeding. Okay, so these are the fluids that contain HIV. And diagnosis: it is diagnosed by ELISA. It is the short form for enzyme linked immunosorbent assay. Okay, it measures the antibodies in the blood to find out the infection is present or not. So ELISA is not just a diagnostic method, it is a method for developing antibodies also. So it is used for diagnosis of AIDS. Okay. Yes, so symptoms of AIDS will be slowly the person will start becoming weak and very easily attacked by diseases. So even fever, cough and cold, and diarrhea, rashes, weight loss. So since the immune system is very weak, person will become really very weak. Okay, so that is about immunity. All right. So prevention is awareness is prevention. Use only disposable needles. Routine screening of blood donors, organ donors, so that their blood should be screened. And even now we have a semen donation, so that also semen should be properly checked whether they are positive, HIV positive, or there is no case of HIV in them. Sexual habits should be changed strict to monogamy, means single partner. Do not share razors. So all these are the basic preventions that should be taken care, so that a person is not contaminated, or a person is not easily affected by this disorder. Okay, so that was about AIDS, and I hope this immunity part is clear. Okay. And in the so in after this you have drug abuse, drug and alcohol abuse. I'll send the PDF of the book itself, so that is nothing to explain in that. Different drugs that are present, the symptoms, and whatever treatment is there, or what are the effects of all those drugs, or adolescence, how people get addicted. So all those general knowledge, or general, there is nothing much to explain. There's point wise it's given. So I'll just send the book notes, books, I'll send a PDF. You can go through, or whatever is given in your book also you can go through. Okay, so go through that part. In this video also you go through. Immunity is very important chapter. Okay, so that is why I won't be sending a video explanation of drugs and alcohol abuse. Okay, so I'll just send the PDF, go through that, and let me know if you have any doubts in that part also. You can ask me. I don't want to prepare a video for that, because that is a self study based chapter. Yes, so that is a common awareness chapter. Okay, so that you can go through. All right, so then now we have one more chapter remaining, that is microbes in the industrial and other uses of different microbes. that I'll do in the next video. Okay, thank you class.